Hello everyone, a very warm welcome to this video. My name is Dr. Sayed Kazmi. Many times in the past I have been asked repeatedly by my colleagues and by my students that why there has been a second and third wave of COVID. It was thought when the first wave came in that after some time, because of the public health measures, especially the hard measures that we had in place like strict lockdowns in different parts of the world, the types of quarantine measures, it was thought that over some period of time we would have this thing under control and this would go down to a level where it would be more of an endemic thing and there would not be much more cases. But over the last year, rather more than a year, we have seen that we have got a second and a third COVID wave. So why? What's causing these second and third waves? So in today's video I will try to answer this question. But before I start, those of you who are watching my videos for the first time or who have joined me right now, please do subscribe to my channel, and if you like my videos, give me a thumbs up and share it with your friends. So let's dive into today's topic. Now the answer lies in understanding the term mutations, and I will explain that in a moment. But let's start by looking at the wave. So I have drawn this sort of a rough diagram which is actually showing the first wave, the second wave, and possibly the third wave. And if you look on this timeline, you would see that when it started somewhere in the beginning of the last year, all of a sudden somewhere in late February and March we had a surge of cases all over the world. So we call it the first wave. So in the first wave we had many cases going up into, the cases ran into millions to start with, and the cases also rose sharply. The mortality rose sharply. But then by the time the summers arrived in July and August, we thought that we had controlled it because the number of cases were on the decline, and it was thought that probably this is going to be over soon. But that around Christmas time or a bit before that, suddenly we saw a sharp rise in the number of cases, so it surged again, and this time it came back with a vengeance. So we saw a lot more mortality in the second wave, and this took the whole world by the storm. So whether it was United States, whether it was UK or the Europe or different parts of the Asian continent, it took us by the storm. And we saw many cases and a lot of mortality. And now we are seeing that in certain parts of the world, especially in some part of the Europe, there is probably a third wave where the cases are steadily on the rise. So probably we are into the third wave as well somewhere. So if you look this timeline, so we have got three waves. Every time it comes down, then it comes back, sometimes with a vengeance. So what is causing that? Now, the scientists who are looking after the COVID, and especially those who are the virologists, they have studied this virus in detail. So the first thing that we know, in the very first wave, we knew that it was caused by the SARS Cov-2 virus, and it was well known during the first wave. But in the second wave, then we started listening about the COVID variants. And three important variants were well known over the world. The first one was the B117 strain, which was detected in UK and then spread in different parts of the world. We also heard about the South African strain, which is the B1351 strain, and the P1, which is known as the Brazilian strain. So most of the second wave of COVID was caused by either the UK strain or the South African strain or the Brazilian strain, and there were a few other variants as well, the California variant and other thing, which were like in certain geographical pockets. So all these variants have arisen as a result of mutations in the original SARS Cov-2. Now the SARS Cov-2 virus, initially it was thought that this virus was actually a virus of the animals. It was more of a zoonotic transmission, and somehow it jumped into the humans. How? We don't know. Probably it has something to do with these live markets in the Far East, especially in China. But whatever had happened, somehow this virus came into humans, and then it started replicating in the humans. So we saw a huge number of cases, and it was given a name, that was the SARS Cov-2. But as the infections, they rose around the world, as the numbers multiplied, variants were seen, and these variants were the results of mutations. So the question is, what is mutation? Why this happens, and how it can lead to different types of viruses which are more resistant as compared to the original one. So for that, it's very important to understand the concept of mutation and how it occurs. So what is a mutation? Let's talk about that. Mutation, simply defined, it's an inheritable alteration in the gene structure. So every living cell has got a genetic code, and we know that in case of COVID virus, it's an RNA virus, and the RNA is a single strand of RNA. So if you look deeply into this RNA structure, so RNA is consists of the building blocks are the nucleic acid. So you've got adenine, guanine, cytosine and uracil. So these four nucleic acids, these are the building blocks of life. These are the building blocks of RNA, DNA, everything. So these four nucleic acids, they can combine in a variety of way, and usually on this RNA helical or in the DNA helical, these nucleic acids are arranged in triplets. So for example, adenine can be combined with guanine and cytosine, then you can have two adenine and a uracil, and you can have a guanine with cytosine and uracil. So you can have different combinations. So these are triplet combination, and each triplet combination of nucleic acid that codes for a particular amino acid. So like for example, I will give example like AGC, like the adenine, guanine and cytosine, let's say it codes for amino acid AA, which is denoted by AA. So then another combination of the same nucleic acid AAU can give rise to another nucleic acid, another amino acid which can be small AA, and then the another combination can give rise to another amino acids, can be another one. So in general, if we reflect back on our knowledge of biochemistry, we have got 20 amino acids. 10 of them are essential, 10 of them are non-essential, and these amino acids are the building block of proteins, and different combination of these amino acids can lead to different proteins. So let's say you have got 3 different types of amino acids over here. Now these amino acids are combined and they are joined by peptide chains, and you can have a long structure which is known as protein. And this protein then springs into life because these are the proteins, these might be the enzymes, these proteins might be the antigens, these proteins might be the the things that actually define the virulence of the virus or the bacteria, if we are looking at the bacteria. But because we are focusing on the virus, so we'll just take the virus into consideration. So virus proteins are formed by combination of by a combination of amino acid, and these amino acids come from different combination of these nucleic acids, and this code is present on the RNA. So the COVID-19 has got a particular structure where these nucleic acids are joined in a particular order, and then these they encode for the different proteins that form the shell, that form the spike proteins, and different parts of that virus. Now let's say if at any point in this code, if one nucleic acid change position, let's say here instead of amino acid adenine, nucleic acid adenine, let's say it is replaced by cytosine. So you have got C here in place of. So what would happen? It would code for a different type of amino acid, or it might not code for anything. So these are changes in the order of the nucleic acid inside the RNA or the DNA. This is known as mutation. So you can have just one mutation, which is known as point mutation, or you can have a mutation in the whole segment where the whole, let's say if these are these 4 pairs, this whole can be replaced by something else, another code. And this is quite common because those cells are rapidly dividing, Inheritedly they have got more chances where this code would be replaced by something. So every time a cell or a nucleic acid divides or an RNA divides, there are chances that it might lead to mutation. In other words, simply mean that some of this code would be replaced by some other form of code. And this happens in all those cells or particles which are dividing very rapidly. So mutations are quite natural, quite natural. Now you also need to understand the Darwin's theory of natural selection. Obviously Darwin was there in the pre-genetics era, but he wrote in his paper that he noted that the species, they evolve over time. So he noticed that mutations or changes in the species are quite common. It happens over time. So some of the species are strong, some of them are weak. So he noted that the weak species in this evolution, they are filtered out by nature. So the nature, anything that is weaker, it would simply die out, it would simply fizzle out. So it will be just filtered by the nature, and those which are strong, they would survive the nature. So we call it survival of the fittest. So obviously all living things, they reproduce. The same goes for viruses. Remember, so the underlying principle here is more reproduction leads to more probability of mutation. So anything that is multiplying rapidly would have more chances of mutation. So obviously when you've got mutations, you would have certain offsprings of that particular particle or that particular bacteria or that particular virus which might be weak, or there might be someone which are quite strong. So obviously the weaker ones, they would not be able to survive, so they will just be filtered out, they will just simply fizzle out. But those particles which are strong, in the sense like they are more resistant to changes, that could be chemical changes, that could be extremes of weather and other things, they would have more chances of survival. So they would be more tougher than their predecessors. So that's why more reproduction leads to more chances of mutation, and that leads to more chances of species or offsprings that are quite tough as compared to their predecessors. And this has happened exactly in case of COVID as well. Now here you also need to understand what is the difference between an antigenic drift versus antigenic shift. So antigenic drift and antigenic shift. Antigenic drift is as over a period of time the virus multiplies again and again. So in every infection, a virus goes inside the human body, it simply replicates. So like 1, 2, 4, 8, and then it goes in another human being, and again it divides and divides and redivides. So as they are dividing and replicating at a fast pace, there will be more chances of mutations, and mutations do occur. So over a period of time, what would happen, slowly and gradually there would be a change in the shape and in the antigenic structure of that particular virus, and it happens very slowly, because with every cycle of replication there are small changes in the antigenic structure and it evolves. So we call it as the antigenic drift. So with every cycle there are changes in the shape of the virus, it may be in the virulence of the virus, and it's a slow one, so we call it the antigenic drift. Then another concept is antigenic shift. Antigenic shift means when there is a sudden radical change in the virus, and it usually happens when one type of virus combined with another type of virus, and a third type of virus is made out of, like, combination of these two genetic materials. So for example, you can have a human variant of a particular virus and an animal one, they can combine, and in that random combination a third deadly form of that virus can come up. That is known as antigenic shift. Now luckily antigenic shifts are quite rare, while antigenic drifts are more common, like the 1918 human influenza pandemic that was suddenly caused by an antigenic shift in the human influenza virus. But the antigenic drifts, it occurs gradually. So that's why, like in case of influenza virus, we see vaccine every year that comes out, so that it incorporates the antigenic changes that occur because of replication every year. So antigenic drift is quite natural. It happens in almost all viruses which replicate quite fast, and it has happened in case of SARS CoV-2 as well. So as you know, it has been replicating in different countries and different races, so we have seen a gradual change in the shape, in the antigenicity of this virus, which again can be very well explained under this heading of antigenic drift. Now the problem is, in SARS CoV-2 we know the spread is it spreads rapidly, and usually most of this spread occurs in the asymptomatic phase of the illness. So before a person knows, like for example, a person who knows he's ill, obviously he's going to self-isolate or he puts himself into quarantine. So what happened, there are less chances of the virus to spread, and eventually it would fizzle out. But when a person is asymptomatic and he doesn't know he's got the infection and he's spreading to other people, so what happens is that the virus quickly jumps from one person to the other person, then to the other person. So it becomes very easy for the virus to replicate. So that easiness of this replication would again theoretically lead to a more probability of mutations. So the more the replication, more the probability of mutation. So we will definitely get mutations over time when a virus is so easily spread and can so easily replicate. Now, what has happened in the first wave, as the virus spread all over the world and different countries and races and ethnicities, they were affected, and it was on a rampage. Because of this rapid replication, there were mutations. Some of those mutations obviously were weak mutations, so they fizzled out. But over a period of time, 3 mutations occurred in such a way that they led to variants of this SARS CoV-2 virus, and these variants were known as variants of concern, because these variants were responsible for the second wave. So they were more transmissible, they caused more severe disease. It was easy to spread as compared to the original one in the first wave. And these 3 variants that we saw were the B117 strain, which is known as the UK strain. It had 18 mutations as compared to the original SARS CoV-2. Then we had the B1351, that is the South African strain, that had 8 mutations. And then the third one was the Brazilian strain, which was known as the P1, which had 21 mutation as compared to its predecessor. So we had 3 variants of concern: the UK strain, the South African strain, and the Brazilian strain, which were predominantly responsible for the second wave. So the question why the second wave came in, the second wave came in because of these mutations we had variants, and these variants made the virus more antigenic, they made the virus more lethal, they made the virus more easy to spread. How? Look at this. Let's say this was the original virus. Here you can see this is the original virus. So you can see these are the spike proteins, which are in the form of let's say a spike with a sort of a rounded end. So the B117 18 mutations led to change in the protein structure, which led to a different antigenic state. So here you can see more of a sort of a what you call a ice cream shaped spike protein, let's say, and we call it the B117. The B135 led to another type of mutation, which led to the spike proteins being triangular in shape. And the Brazilian one, the 21 mutations led to changes in the structure of the proteins, which actually translated into a different antigenic structure, into a different spike protein, which was more like, let's say, having 5 or 6 and just like a star. So what happened? The initial, if you look at the initial ACE2 receptor, so this is the shape of the ACE2 receptor. So obviously it just looks like a keyhole. So in order to put a key in, you need that the key fits in the keyhole. So let's say the initial one, when it had the circular, it could fit with some difficulty inside, and then the whole thing was internalized. But when you had this types of structures over here, let's say a triangular or something like a star, so they can more easily fit into this structure. So as compared to the original SARS-CoV-2, now you have got variants which have got spike proteins which can more easily unlock this keyhole, which can more easily fit into the receptor as compared to the original one. So let's say in the original one, if out of 10, let's say 2 receptors were open by then, here out of 10, 8 receptors are open. So you get more internalization of the virus particle, which leads to more severity of the infection. So easy transmissibility and more lethal, because more virus particles can be internalized by the cell, and that leads to more replication. So this was responsible for the second wave, and in certain cases it's also responsible for the third wave as well. So to summarize this, as compared to the original SARS CoV-2 virus, these mutations, whether that was the UK strain, whether that was the Brazilian strain, whether that was the South African strain, these mutations led to changes in the protein structure, which led to change in the shape of the spike protein. Now the spike proteins were in such a shape that they could more easily fit into the ACE2 receptors, which are the receptors on the cell, which are present there in the throat, they are present in the lungs as well, and where this virus could simply attach, and once it attaches with this receptor, then the whole thing goes inside us. That's how it causes infection. So it was more easy now, with the virus being easily, its spike protein easily getting fit into the AC receptor, it could easily be internalized. So it means more transmissibility, and it could easily infect the cells, and that led to increased severity of the illness, because now you are opening more keyholes and more virus particles are going inside, and more replication, and your body is overwhelmed with virus particles, so much so that your body defense mechanism cannot cope with that, led to high mortality. So these mutations were responsible for changes in the protein structure, in the spike protein shape, making it more easy for the spike proteins to attach to its receptors, get internalized, and cause infection. Now the question is, what are the mutation possibilities and what can it lead to? And for that we need to understand that there are 3 groups or 3 headings under which the mutations can change the scenario. Number 1 are those mutations that increase the transmissibility of SARS CoV-2 virus. So for example, this is a person who is infected by the virus, and he is let's say he's coughing and he's spreading out the virus. Now this virus, as it replicates in different people, here from the original one it changes to a more virulent form, because the mutation has caused a new thing to come up. And let's say this can more easily fit into its receptor, as I discussed earlier. And what happens when it can easily fit into the receptor? It can more easily go inside the cell, and that means more quick spread, and more quick spread means high number of cases, and when there will be more number of cases, that would lead to high mortality. And this is exact what has happened in the second COVID wave. So that 3 mutations especially, and there have been other mutations as well, but these 3, the UK, the Brazilian and the South African strains, they actually spread more quickly. They said that the transmissibility is 30 to 50% more as compared to the original strains. Number 2 is those mutations that lead to avoidance of the host defenses. Again, let's say a person is infected and he is coughing out the virus particles. Now these virus particles, let's say when they go inside your body, like another person is infected, so what happens in the case of the original ones, your body develops immunity against this, your body develops antibodies. So these antibodies, how they actually recognize the antigen which are the spike proteins, and they actually combine with that, and then they neutralize. So once the antigen antibody complex is formed, then the macrophages, the neutrophils in your body, white blood cells, they can simply filter them out, they can simply destroy it. But let's say when there was change in the antigenic structure leading to a different types of spike protein, now what happens? You've got antibodies already. Now these antibodies are against a different type of spike protein. Now you've got a different type of spike protein. So what happens? Your antibodies, which are already there in the body, they are not able to attach to that. So if they are not able to attach, they cannot neutralize these new variants, which have arise as a result of mutations. So when your antibodies cannot neutralize these viruses, it means they are more virulent. They can easily, go anywhere in the body, affect your organs and cause damage. So there will be more spread, there will be more organ damage, there will be high number of cases. Again, that would translate into a high mortality, and this is exactly what we have seen in the second wave, and also we are seeing that in part of the third wave as well. The third is those type of mutations that can cause a virus to avoid vaccination defenses. Now we know that vaccines are available. Many countries have started rapid vaccination drives. In UK, 50% of the adult population is already vaccinated. In Israel, they have vaccinated the majority of their population. In European countries, they have just begun with the mass vaccination. So it was initially thought that vaccination would lead to control because it would lead to herd immunity. But again, we don't know. As long as mutations are there, we don't know what would be the final shape of this virus, because again, a person has got this virus, original virus, and this original virus is replicating, freely. So again, because of mutation, the original virus mutates into a mutated form, again with a different antigenic structure, antigenic spike. Now what happens? Let's say you have started vaccinating the public. So you've got different type of vaccine. You've got the Pfizer and the Moderna, which are the messenger RNA vaccine. You've got the AstraZeneca, which is the DNA vaccine. You've got the kill vaccine, like the Chinese vaccine. So what happened? These vaccines, when you administer them, so either the RNA or the DNA, it enters the human cells, and it allows the human cells to form parts of those spike proteins, and those spike proteins are released in the blood without the virus. With the total, you don't have the total virus, you just have got these spike proteins. So antibodies are formed against that spike proteins. So whenever the actual virus comes into your body, these antibodies, which have already been formed, by introduction of this vaccine, they would attack and destroy the cell. So it seems quite simple. So what happens? But if you have got a changed mutant virus whose spike proteins are different, so you've got antibodies against a particular type of spike protein, but now the spike protein is changed, the shape is changed. So you have got these antibodies which have been formed as a result of vaccination. They are not able to fully neutralize them. So some of them would be neutralized, but still many of the viral particles, they would not be neutralized. So you will still get the disease. Some people say you'll get the milder disease, and some, if you are, let's say you've got immunocompromised, or if your immune system is not properly working, they can overwhelm you. Similarly, if the antigenic structure is totally changed, even if you've got good level of antibodies, they may not be able to stop the infection. You might get infection, you might get severe infection. So that's why the crux of the COVID control is on reducing the number of mutations. And how would you reduce that? Because mutation is something which is quite natural. Anything that replicates can lead to mutations. So that's why the replication has to be reduced. And how? Again, by reducing the number of cases. And how would you reduce the number of cases? Again, very simple things like they say use of masks, proper hand hygiene, maintaining social distancing, and some other measures, like if somebody is thought to be having this self-isolation and things like that. So that would reduce the replication. The reduction in the replication would lead to reduction in the chances of mutation. So how it will shape in the future, we don't know. Maybe over a period of time we might see a few waves coming in. And there is also a high chance that it becomes more of a seasonal thing, because as the winters they approach and people get more indoors, there are more chances of not maintaining the social distancing. The people will be very close to one another, and again, because of the closed atmosphere, closed environment, it's more easy for the viral particles to jump from one person to the other person. That will lead to, again, replication. It might lead to seasonal exacerbations, and over a period of time then it might come down from the pandemic level to an endemic level, where you see a constant presence of the virus, but it's not like too many high number of cases, but you get cases here and there. So there is a constant low presence of the virus, because I don't think this virus is going anywhere anytime soon, because the number of cases are so high. The reservoir of infection is still there, and it's a human body, and as long as it is replicating, it can spread. And as the more fast the replication is, the more the chances of mutations and leading to change in the structure of the virus, in structure of the spike protein, which makes it more virulent, which makes it more transmissible, which can make it more dangerous or lethal. So to summarize all this, why the second and third wave has occurred, it's simply because there were mutations from the first wave, and those mutations led to some of the variants which were known as variants of concern, the UK strain, the South African strain, and the Brazilian strain, which were responsible for many cases, surge in cases and also in mortality, which was caused in the second wave. And where it didn't reach, it is now causing the third wave as well. And there are fears that there might be more mutations in the future. And that's why the crux of future control of this would be investing in vaccines. We can't simply be happy that we've got 2 or 3 vaccines. These vaccines need to be constantly redeveloped. Again, as the variants are coming in, so these vaccines have to be tailored according to the new variants coming in to make it appropriate for the new types of viruses. So if the companies which are working on this vaccine development, if they keep on that work, constantly modifying their vaccines, then we might be able to bring it down to acceptable levels. So the future scenario is, number one, we know that the virus is constantly mutating, especially in the younger population, and so far this younger population has been kept out of the vaccination drive. So obviously they don't get severe infections, but they are responsible for asymptomatic transmission. So the reservoir of infection is there. So we need to have strategies for mass vaccination. So somehow we also need to develop vaccines to cover the younger population. Only then we can think about decreasing these mutations and bringing the thing under control. Number 2 are that seasonal peaks are expected in coming 5 years. So obviously, as I told you, it's going to stay with us for some time. So seasonal peaks, especially in winters, November, December, and January in the northern hemisphere, and May to September in the southern hemisphere, are expected. And as I said earlier, vaccine development needs to constantly adapt to new mutations so that the vaccine is effective against those mutant strains that are arising every now and then. So only these 3 things, if they are taken into consideration, we can think of the future in which there would be control over the COVID spread. Otherwise, we might be seeing these peaks coming and going, and we might see these lockdowns happening every now and then. So that was all about the spread of COVID-19, especially in context of the second and third wave, why it is happening. I hope you have understood this thing. If there are any questions, you can put your questions down in the comment section below, and I will answer them. So have a good day. Bye bye.